So let's get started. So the learning objective, we already discussed our circulatory system consisting of circulating fluid. Of course, a lot of times we will be discussing more of a blood vessel, blood, but at the same time, we will also know that there's a lymphatic flows, which is also circulating fluid. Circulating is a little bit weird because lymphatic flow is a one-way direction from the tissue to, in fact, at the end, it's into the heart. Okay, so that's a one-way, that's a little bit different. And a system of blood vessels and a pump, okay, and general structure of blood vessels. So this one I already showed you in the video just for you to appreciate. So this is the video I collected. So this is my own data, and you can see a beautiful blood vessel from top of the mouse. Cerebral cortex is going down. What's striking here is you see the beautiful distances between these capillaries. So this actually shows that the importance of diffusion from the capillaries exchanging oxygen and nutrient between all the cells inside in the brain tissue. Of course, there are lots of neurons and neuroglial cells which requires all this communication from this blood vessel. So the structure of the vessel, this is just exquisite. You see the distances, a beautifully distances one. And I do not actually know exactly what the mechanisms are, but it's just an amazing thing. So regarding the blood vessel, which is very relevant to tumor biology and our top killer in advanced countries for tumor, and we know that the tumor needs a feeding, and that's coming from so-called angiogenesis, a new vessel formation. And there's a kind of new science paper in 2016. Of course, I think this concept could come even a little earlier, but they made a nice distinction of a new vasculogenic mimicry. That's kind of very interesting, that angiogenesis versus vasculogenic mimicry. So I think you already know what that is. So let's get into directly to the question of Wuzhu. Where's Wuzhu here? Let's see. Oh, yeah. So Wuzhu is asking about what's the difference between angiogenesis and vasculogenic mimicry in kind of tumor or procedure of making. So this is a little confusing question to me, but anyway, Anho also made a good point here. So Wuzhu, can you explain a little bit more of your question? Because at the end, this part is a little bit unclear to me and maybe to others as well.
In the slide, there are two ways to feed the tumor, as we see. So I just wonder about how to different the procedure. I mean, angiogenesis and vasculogenic mimicry. Is there any different kind of tumor?
Yeah. Oh, yeah. Okay, I see. I mean, this is a very valid question and very important question, in fact. So why we do care about like angiogenesis and vasculogenic mimicry? Why do we care to distinguish between these? And Wuzhu is asking about what kind of tumor. In fact, honestly, I do not know specifically which tumor kind will prefer to have these. So this is an excellent question in terms of research quality. And so I think that's interesting. So Anho also find out some. So Anho's finding is about kind of a tumor-induced vessels are sinusoidal, meaning that tortuous. So vessels are look more like a little bit like wiggled, okay? And even this is important point. This continuous basement membranes, okay? So where's Anho over there? Oops, I can't see Anho. Somehow she has an issue today. So why this is important? You know that the vessels have endothelial cells directly facing down to luminal blood side, and they have their basement membrane continuous typically. So in tumor, this continuous basement membrane, which is very much indicative of the important abnormality of tumor blood vessels. This could be utilized for tumor cells to metastasize, getting into the tumor, or our targeting agents or our treatment may take advantage of this. So this is something good to know and important, and also lack tight endothelial cell junctions, because endothelial cells has to have tight junction to seal inside the vessel, and somehow in tumor tissue, even this is compromised. So that makes it hyperpermeable to macromolecules. So this is an interesting point made by Anho. Why? We call this as a leaky vessels. So what are the macromolecules inside the vessel? There are many proteins, including the biggest one, albumin, and they leak out this outside of the vessel. So what happens if macromolecule goes outside? And you can think of in terms of osmotic pressure. So to keep the osmotic pressure, the inside water has to leak out to keep the concentration higher, lower than before. So that makes interstitial fluid pressure high. This is one of the reason why tumors are solid and tight, more stiff. So that's one of the reasons. And the problem of this is pressure in the tumor tissue is higher. Your chemical treatment or your drug cannot reach into the tumor well because the pressure is higher. So this is a very important aspect of this. Let me see. Yeah, I think most resulting areas of hypoxia and necrosis. So this is, while we have another like whole week of tumor session, this is already kind of alluding to that, hypoxia and necrosis. What it means is in tumor, which is of course abnormal organ-like structure in our body, and they suffer from this abnormal blood vessel, which make a local hypoxia, meaning that lack of oxygen, because the blood vessels are abnormal and not functional very well. And so sometimes there's especially center of the tumor we call necrotic core. So core is necrotic, meaning that they are suffering from the hypoxia. They don't have enough nutrition, so tumor cells die inside. So this is kind of also typical. So hopefully that's giving some sense. So I think I also look for from Google search that regarding to Udu's question, because this is such a very interesting topic. In fact, you can't find it in typical textbooks. So that's why I love this question. So vasculogenic mimicry, what is vasculogenic mimicry? So it's in fact, there's no vessel here, but it looks like there's a hallway here. So it looks like vessel-like. So that's why mimicry is there. And because it's a vessel-like and vasculogenic, and there's a terminology for vessel formation. There are two terminologies. One is angiogenesis, like here. What does angiogenesis mean is that like tumors take that strategy. Tumor love this nearby vessel, already existing vessel. They secrete like growth factors. So for example, VEGF, vascular endothelial growth factor. And they triggers the new vessel forming to feed into the tumor. So that's called angiogenesis, a new formation of vessel from existing. But think about your development when you are nothing and then you start to form a vessel that doesn't have an existing vessel. So you have to create vessel at the beginning. So that's called vasculogenesis. Okay, it's literally vessel genesis, generation from out of nothing. Of course, there's a so-called HUVEC cells, a human umbilical cord endothelial like cells. You guys know from some of your literature. So that vasculogenesis is kind of mimicked by the tumor. All right, so let's go back to here. So what is vasculogenic mimicry? Is the ability of cancer cells. So that's important for cancer to organize themselves into vascular-like structure to get nutrients and oxygen independently of normal blood vessel, because there's no normal blood vessel inside tumor, or sometimes they without angiogenesis. So why Uju's question is important? Because, if you know how tumors survive in our body as they form tumors, if they adopt angiogenesis, you block angiogenesis, you develop a molecular trick or treatment to block angiogenesis. But what if the tumor develop non-angiogenic way of feeding themselves, for example, like this vasculogenic mimicry? Then your treatment of anti-angiogenic therapy fails. So that's why if you know how vasculogenic mimicry, how the mechanism occurs, you can come up with a new treatment of blocking vasculogenic mimicry. And I hope this one makes sense to Uju and people. So yeah, that's something over here. You can find a number of literatures, but unfortunately, I couldn't find a major source of vasculogenic mimicry in terms of a specific tumor types. But there are a number of very interesting studies. You can see vasculogenic mimicry in carcinogenesis, cancer formation, and clinical. In a hypoxic tumor, you see the mechanism, why the mechanism study is important. It's even like last year, 2 years ago, last year, with a high citation, so that you can see people are pretty much interested in advanced research. And there's a big high citation over here. And so as I said to you, targeting vasculogenic mimicry by phytochemicals, and even platelet disrupt vasculogenic mimicry by cancer cells. So these are kind of a basic research, but that's interesting and important in terms of when we develop a new treatment and better treatment, which avoid the current studies and current ways of treatment. Okay, let's move on to human circulatory system. I guess you've already seen, but I just want to point out that this kind of diagram, you can see the liver, very interesting, hepatic vein, and there's a gut, there's a portal vein and hepatic artery, mix them together inside the liver and through the liver sinuses, and they drain out to hepatic vein. So now let's get into another question of vascular circulation. So where is it from? Oh yeah, there it is. Why is the artery bright red while the vein is dark red? Okay, very important and interesting question. And Doreen also tried to answer here. So let's say I want to ask you a question because you asked this interesting question. If you look at here, you see my surface vein. How does the color look like? Is it clear?
I think it's dark red. Right?
Dark red? Is it? Youngmin, I want to see your face. Youngmin, what's your opinion? From show tell this is dark red. Youngmin, is it clear?
Yes, Professor. Sorry.
Yeah. What's the color to you? The blood vessel color. You see, is it clear? Not clear?
Green?
Oh, yes. Yeah, sometimes it looks like green or blue or purple. So let me ask Youngmin a question for this. Why do you think blood vessel, especially its vein, looks green to your eye?
I think vein blood deoxygenate and several epithelial cells.
You don't have to like this is a little more open. You have to think, right? So you said it's a deoxygenate blood. Very good point. So let me ask you the question. Deoxygenated blood is a green color or blue color? What's your thought?
I think red color.
Red color?
Yeah.
Okay. I mean, this is very interesting question. And in fact, I found no clear answer even on the web. That's why this is something very interesting. I would say if someone who finds a very clear explanation, very logical, very reasonable, legitimate, I would give a double points for this, okay, in YouTube. All right, so there's a Dorin also give a answer, which is good. Let's see. Chemicals appear particular colors to our eyes. Good. Based on wavelength of light they reflect. Very good point, Dorin. Where's Dorin? Okay, there is Dorin. So hemoglobin bound to oxygen absorbs blue-green light. So this is oxygenated hemoglobin is absorbing blue-green light, which means it reflects red-orange light into our eyes. That's very true. Good point. So that's why blood is always look red. Okay, and blood turns bright red when oxygen binds to its iron. Without oxygen, blood is dark red. So this part is, I think it's a good attempt, but it's a little bit not too clear. So that part that we want to get into a little bit into. All right. So let's see. First, for that, I think it's important from you to think of the spectrum point of view. Okay, so why blood is red? Because the color comes from hemoglobin, which absorbs green and blue. So that's why red color is only reflected to our eyes red. And why oxygenated blood looks brighter red and deoxygenated looks a darker red. The reason is we should be finding in from the spectrum of absorption here. So this will be actually repeated coming in again in the later part of our class in biomedical optics, but it's actually a good idea to see here. So look at here. So you know that the visible light is coming from 400 to 700 nanometer, and this is absorption curve. So look at red and blue. This is red stands for oxy, blue is the deoxyhemoglobin. So from the blue and green, they look almost similar, okay? So they absorb more light of blue and green, so that's why, and lower in the red color region, that's why blood looks red. But oxyhemoglobin has a much lower absorption, meaning that a lot more reflection for red color. So that's why it's a bright red, while deoxy has a little more absorption for the 600 to 700 red color. That's why it's a more darker red color. So I hope this one makes sense for you. But the real question to you is, in fact, why this superficial vein looks actually bluer or even green color. As Youngmin says, green. That's also possible. And this is something I haven't found a very clear answer to you, but this is more like interesting. So here's one thing, answer, blood is red, bright red, and vein, because there's no oxygen, blood is darker. So that's what we saw from the spectral point of view, okay? And there's an interesting another point, why veins are darker, that let me tell you, even if there's a Google here, it doesn't mean that it's always true. So you have to be critically see. Veins are darker than arteries because they have thin walls, okay? And the darker blood shows through them more easily. Arteries are lighter in color because they have thicker walls containing connective tissue, which is lighter in color in preserved material. I personally do not agree with this because It's true that veins are thinner, but arteries are thicker. But that doesn't necessarily make their color changes. It's possible the scattering will be different, but I personally do not agree with this. So you look at that, the source of this, and you have to judge yourself, okay? The truth doesn't always mean that it's in Google. All right. And the other thing, heart pumps the oxygen-rich blood is then pumped out. Okay, so that's why you know that bright red color. And the color of vein. So people say venous color is dark red. And as I asked Youngmin, that this green color, it appears to our eye. Doesn't mean that the blood is actually green or blue. And the reason is probably a complex. It can be a little complex through the interaction of the light, visible light getting inside the blood vessel and coming out. There's a tissue, skin color, and that scattering and absorption all combined to reach to what we perceive our color of the vein. Because arteries are deep inside, so you can't really see the arteries quite easily. Okay, so you know, veins are types of blood vessel. So many people wonder why veins look green or blue through the skin instead of red. I give you a chance to really find this out. As an answer in YouTube, I'll give you twice more points for that because I haven't found a very satisfactory answer for this. All right. The other thing is there are some theories here why my veins are green. As Youngmin said, green veins are superficial veins, right? That's what to our eye, near the surface of skin. They appear green instead of red because green has a shorter wavelength than red. We know that green is shorter wavelength, and shorter wavelength has a more scattering in tissue compared to red. So red color gets deeper inside compared to green color, but it doesn't really show why blood vessel versus the other skin scattering skin different. So that's why it's still not that satisfactory. You are more likely to see green or bluish colored vein if your skin is lighter color. This is very true. So if you have darker skin, because darker skin means you have more pigment, melanin pigment, and the pigment has a high absorber, and it's harder to see the color of your vein, right? And why veins blue? So there are theories here. Veins appear blue because blue light is reflected back to our eyes. Very true, but it doesn't necessarily distinguish blood vessel vein versus other tissue. So that's why these answers are still not very specific and satisfactory. So hopefully I'll expect you to actually answer a little deeper into this, okay? This may come to your exam as well, actually. All right, so let's move on. So structure of vessels, because you guys all know here, so let's move on. Hamza. So yeah, so veins are thinner and arteries are thicker. So in anatomy, artery has a typical 3 layers. So we call intima, media, adventitia, or intima called interna, media, externa. Okay, so intima is the innermost. So it consists of exactly endothelia.
Sorry.
No, that's no problem. Epithelium is outside facing. Endothelium is in the blood vessel or lymphatic vessel. So that's endo. So that's a little important. And media is a middle layer, is composed of muscles and elastic fibers. Hamza again, can you guess what that is? This is only artery.
I did not revise yesterday videos.
Okay.
Sorry for that time. Actually, I was busy.
Okay, that's fine. Okay. Then let's move on. Soyeon. I mean, usually the answer is in the slide, so don't worry too much.
skeletal muscle.
Great. Yes, smooth muscle cell. Smooth muscle cell itself in general has no direction unless you have make a directional ones. So smooth muscle contract, and artery is a thicker, it contains a muscular layer because it has to sustain a pressure and it has to be elastic. Okay, so let's move on to the external part. Adventitia. You know, these terms are difficult at first, but you have to know that. So that's why you get to familiarize. Tunica adventitia consists of fibroblast and 신소연 again. 소연이?
Yes, Professor.
Yeah.
Collagen fibers.
Exactly. Yeah, collagen fibers. So collagen is a very important in terms of holding, especially a tension, okay, resisting tension. So that's a collagen fibers. All right. So structural vessels, very, very important. One thing you make a note is veins are much thinner because they are not necessarily responding to pressure because the pressure is way lower. And there's because of that, there's a valve to prevent backflow. So here is the thing, structure of the vessel, which we already told you. Yeah, so here, artery, vein, and capillary. I put this very interesting tables showing the structure and the actual diameter, and even relevant tissue makeup. So one thing I point out here is, let's see, I think, yeah, we have many slides, so let me move on. So this is inside tunica intima, okay? And the middle layer, tunica media, because it's a middle, okay? It's a smooth muscle. And outside, adventitia, okay, that's collagen fibers. So what matters here is really about the mechanical or physical point of view of the vessel. So, you know, you don't want your vessel to collapse all the time because blood has to flow. So looking at this vessel, what makes a physically blood to flow? And first of all, this pipe, there gotta be a pressure difference, or we call pressure gradient. So higher pressure to lower pressure, it flows. You guys know that. And what about the vessel wall itself? So vessel wall itself has to expand somehow. Then the inside the vessel, so-called like vascular pressure, versus outside of the vessel, interstitium, inside has to be higher than outside to make the vessel inflated. So that's important. So let me ask you a question. When does the vessel collapse? Okay, then this outside pressure is higher than inside the pressure, it can collapse. Okay, so let me ask you a question. In our body, do we have that happening? Outside of pressure is higher than inside? Raise your hand. Where do we have this outside of interstitial pressure is higher than inside? Do we have in our body? Raise your hand. No? Okay. Let me ask you another question. Then what about in the tumor, let's say, in pathological condition? Let's say in tumor, does this happen that interstitial pressure can be higher than internal pressure? Raise your hand. I mean, this is your guess. Yes or no, okay? Yes, can happen. Okay, no, raise your hands. It will not happen. I mean, this is either or the other, so you have to participate in either way. Okay, again, yes, can happen. Okay, nobody. Okay, no, then you guys are all saying, no, it will not happen. Raise your hand. Okay. Okay, what about those who haven't raised their hands? Okay, Janice. Janice, I want to hear your logic or your opinion on this.
I have no idea about this, but I think
Yeah.
this can happen. Maybe I don't know.
Then you should have raised your hand that can happen. Okay, so let me tell you the answer. The answer is yes, it can happen, and what we see is in tumor, you know, tumor cells are like kind of exploding. They keep grow. They put pressure on nearby vessels. Okay, so in fact, we found a clue that tumor vessel collapses a vessel, so it blocks the vessel. And what can happen? You know, you cannot deliver your anti-cancer drug to the tumor because tumor cells collapse vessels. You know, this is 2004 Nature paper. Okay, that's why you have to ask this question and then prove it, and then realize and show why this is clinically important and what are the mechanisms and how we can actually reverse it or change it. Okay, so I mean, this is a simple, not simple, but you know, just based on curiosity too. So you have to consider this, but not even tumor. What else we can have this condition is actually relevant to cancer treatment. For example, breast cancer patient in the past, you know, we have this breast, there's a nearby lymph node. So that draining lymph node, the first one we call sentinel lymph node. The surgeons come in, if you find the breast cancer tumor here, the draining lymph node, the surgeon just took out all of them in the past, okay? This is not even like a decade ago. What happens? If lymphatic is disrupted, then your drainage, tissue drainage is disturbed, and you can't drain tissue, the flow. So the buildup of the fluid makes your arm here and here, this side of surgery, your arm become like twice. And you have a lot of pain. And what happens is with this high interstitial fluid pressure, they collapse the lymphatic vessels as well, and it can't drain. So this is very important aspect of why we have to understand this physical aspect of blood vessel physiology. All right, so for that, we have one formula over here that C, or compliance, is the ability of volume change with respect to pressure changes. Okay, so for example, you guys know from the heart, left ventricle pumping. The first artery is called aorta. So aorta holds the highest pressure differences. Even the highest pressure it has to resist. So that's where we call pressure reservoir. Okay, so that's a compliant, highly compliant aorta. Let's say the other part is a vena cava. Vena cava is a big vein right before getting into the heart. So the vena cava can have like this very big diameter. It's a whole drainage. And that's a volume reservoir. What it means is it has a high compliance, meaning that even small pressure difference, it changes its volume a lot. Okay, so hopefully that makes sense to you. Let's move on to the relationship of the blood vessels radius, resistance, pressure drop. And I just want to point out one point that, let's say our water is 0.75, like this is a viscosity unit, centipoles, okay? Then we are curious of what about blood? Our blood is about 3, which is about 4 times. So you can think of our blood is 4 times more viscous than water. Of course, I think depending on people's age and condition, especially like when people are getting elderly and having a more problem of their blood composition, and you will have a finding a different viscosity of blood. So in fact, one of our faculty members in GIST developed a device to measure this viscosity, and it's also commercialized. Okay, so this is could be an important parameter of finding the state of your blood in terms of viscosity. And in comparison, olive oil is 81, so that's huge, almost like 100 times more viscous than water. All right, so blood is a suspension of RBCs in a protein-rich fluid we call, let's see, Yundami.
Yes, Professor.
Yeah, the fluid in the blood.
Protein-rich fluid plasma.
Exactly, yes. And the point over here is blood is not just a fluid. It's we call multi-phase fluid. Why? It's not just simple or liquid. There are actual cells inside. Even more than 40% are occupied by cells. Majority of them are red blood cells though. So in fact, their mechanical in terms of fluid-like behaviors are quite not easy. I mean, it's complicated in fact, right? So, and one of the part of parameter is a viscosity. All right, let's going on to here. Oh, oops. I'll move on to where? Okay, the next slide. So Ohm's law applied to fluid flow and hemodynamics. So we call this blood flow inside our body is called as a hemodynamics. Hemo means blood and dynamics is a dynamic change or changes over time. And here, interesting question was also arising with the capillary diameter, because capillary is the business end of our blood circulation. And it's very, very important and still very curious as well. And the consequence of branching vessel network, because it branches out, that means it will be smaller and smaller diameter from the thickest aorta, biggest diameter from aorta, which could be over 2 centimeter, 2 to 3 centimeter diameter, to artery and getting down arterial and down to capillary. And so like the important question to ask is, so our body cells, how close they have to be nearby capillary? Because in the end, capillaries provide the nutrient and oxygen by simple diffusion. And of course, there's some convection inside. But so even every metabolically active cell in the body has to be within some distance of nearest capillary for adequate nutrients for metabolism. So, Tami?
Yes.
Can you guess what would be the necessary distance?
100 micrometer.
Very good. This is an easy number to remember. It's 0.1 millimeter. So your active cells have to be within 100 micron. So you can ask whether this number is how like kind of accurate this number, then I can answer no, not very clear. But from next week, we can actually explain where comes this number. And in fact, what recent study of mouse brain, the whole mouse brain, take out the whole capillaries and finding out the distances between the capillary, it actually is shorter than this. It turns out that like 50 or even like dozens of micron. And you can guess the diffusion distance is more like exponential decay. So you have to be within. The brain is known to be the most active cells in our body, brain and heart. So you can guess and imagine their distances from nearest capillary has to be shorter than 100 micron. And this is a little bit of an equation. You can see Q is flow, so it's more like analogy as a current. And there's delta P is a pressure difference, which is driving force. So electrical analogy, you can see as a V. V equals IR or V equals I, V times 1 over R. So this is relevant to resistance. That's why this parenthesis 1 is actually 1 over resistance. And one thing you have to point out, you know, like if someone asks, a baby or kid asks you about, hey, daddy, or hey, uncle, what is the resistance if your blood vessel get down to be half of its diameter? What would be the resistance? Then you can actually proudly say, hey, that resistance is related to 1/r to the, let me see, Janice?
R over 4.
Yeah, 1 to the 4th. So if you have a half of the blood vessel narrowing, you have 16 times higher resistance. What does that mean? So resistance is 16 times higher, then you have, it's more difficult to perfuse 16 times. Or to make your current or flow keep the same, you need to provide 16 times higher pressure difference, which is of course not that easy. So your heart will start to pumping when you have this problem. Try to keep the flow more normal. Okay, so so for that, this is a way our body to control the blood flows here and there. So local blood flow can be controlled by constriction and dilation of these arterioles. Why arterioles contain smooth muscle, having vascular walls delivering blood to the tissue. Then you have to ask too, hey, is that true? And why? This is an active area of our current research. So look at this Nature paper for capillary. Look at the title. You see, if you read the title, you can think of why this is Nature. Of course, you have to provide enough of an evidence, okay? Look at that. Let's read. Capillary parasites regulate cerebral blood flow in health and disease. Oh my gosh. You know, in the textbook, it said that arterioles will constrict and dilate to control blood flow. Why? Because arterioles contain smooth muscle. So you can use a staining called alpha SMA, smooth muscle cell staining, to show that, hey, arterioles are the one who keeps track of and control the local blood flow. But here you say, hey, yeah, okay, yeah, that may be true, but we found parasites, which is another, which I didn't tell you about this, but all these capillaries are in fact covered by parasites, which is along this blood vessels. And unfortunately, they don't contain smooth muscle cells. So this is why people do not think of parasites will not regulate blood flow, but this paper actually showed the evidence. And if you look at the paper, this is only like kind of ex vivo study. So still, it's not that clear, but you know, always after this controversy over time, we start to establish our knowledge base, okay? And then you can ask, why is this important? Yes, this is a more basic question, but in terms of the most devastating disease in modern world is all vascular origin and vascularly relevant. So that's why these are such an important, actually even interesting. So Youngmin, you ask a question. It's a good question, I think, telling white blood cells are about dozens of micron and RBCs are 7 to 8 micron. And the capillary diameter is, you know, depending on the source. Okay. But how can leukocyte pass through the capillary? Yeah, there's a little type of pressure-driven blood flow. So, Youngmin, I want to point out that, in fact, it's known that RBCs are typically comparable, but actually bigger than the capillary diameter. So what you wrote it here, you found in some sources, it may not be always true. So let me tell you the difference. And the other thing I want to point out, in mouse capillary diameter is in fact not 8 to 10, it's about 4 to 5 micron diameter. Okay, But mouse red blood cells are actually bigger than this. It's still like 7 micron. So think of even capillaries. How come RBCs can crawl through this capillary? It's very, very interesting and very important phenomenon. That's one thing. And also, I like your question because I also asked the same question. Okay, I still do not know the answer clearly, but this is such a fascinating question. And why is it important? There's a paper in 2019 in Nature Neuroscience claiming that, you know, Alzheimer's disease, it's known Alzheimer's disease, it's known that the cerebral cortex blood flow is down by even up to 25%, which is a lot. So if you have a dementia with Alzheimer, you have a lower blood flow in your brain and you're suffering, whether it can be cause or it can be a result. We do not yet know clearly. One of the reasons why they claim is, I'm talking about it's an animal model of Alzheimer's disease. They found exactly what you say, leukocyte got stuck in the capillary of Alzheimer's disease mouse brain, and that decreases blood flow. So what they claim is, hey, this leukocyte blockage of capillary, we call this a capillary stall phenomena. They claim this is an important relevance to the Alzheimer's. And you know, with that, you cannot publish in Nature Neuroscience. So what they did is they deplete the white blood cell by putting an antibody against this white blood cell. So they remove this leukocyte and then show that blood flow enhanced in the mouse brain. And that's not enough. They even show cognitive recovery. So they make this mouse run experiment to measure their cognitive ability and the cognitive function restored. So this is striking. That's why it's Nature Neuroscience. I'm not telling that this is happening in the human as well. It's still controversial, but that's why these kind of questions are so important. Okay, so I also took some of this one. So just to make sure that everyone's on the same page, you know, we know veins are generally large in diameter. We know that. And there are different. Okay. Yeah, someone's enjoying music. Maybe volume down. Okay. So yeah, we all know arteries and veins. So there are differences, but I want to point out here that the size, capillaries are very small and will not be easily detected. Right, that's why we cannot see capillaries with our eyes, so small, and they're the same. And look at that, the vein has some diameter. So I think this is, let me see, is this relevant to Youngmin's question? Yeah, I think that probably here's more relevant. Okay. So as regarding to Youngmin's question, are capillaries and RBCs the same size? The answer would be yes, I think they are comparable. Okay. But one point of Youngmin's question is some capillaries are slightly smaller in diameter than blood cells, forcing them to distort their shape to pass through. So this is why our RBCs, fresh RBCs, mechanical property is very important, such that they can actually, like this, you know, this disc shape has to be like this, and then pass through the capillary. They have to change their shape. So old RBCs become stiffer, they can't do this. So in our spleen, we actually sort them out and then remove them in our circulation. Okay. So look at this. RBCs bigger than capillaries. We found, or they found, RBC width is about 25 larger than capillary diameter. So what do they implicate? So the flexibility of RBCs are important in our health. And also the capillary has to have enough of resistance of the strength for the capillary not to rupture if RBCs are like hard. And one thing you guys will be good to know, RBC versus white blood cell as you look at that. 6 to 8 micron is RBC, like, or 7.5, or white blood cells are in general bigger. So Youngmin's question is very valid. Hey, our capillaries are small, and how come white blood cells pass through? And it's still, to me, it's still kind of a mystery. I did the imaging with looking at white blood cells actually rolling along big blood vessel, and they get stuck in the capillary. Somehow they crawl out. So another point of this discussion is some white blood cells, the most hard part is their nucleus. And some white blood cells has nucleus, not just like this red big jump, but they have this lobules like beans, okay? So lobular. And my friend was actually asking this question and using 2-photon microscope in vivo to see why do they have this lobular nucleus. And his explanation is this white blood cell, so including immune cells, their role is inside circulation. They have to crawl outside of the tissue to go to the inflamed area. For that, you can't have just a big nuclei, but lobular one, so that you can crawl out in between the endothelial cells to get into tissue more easy. So it's a quite interesting explanation, but I'm not sure how to prove it. So that part is a little harder problem. Okay. And then one thing, so Youngmin's question is a little general, that he said white blood cells are 12 to 17 micron. But we have to go down to a little bit, because RBC and WBC, RBCs are just one kind, but white blood cells have various different kinds. So it's actually good to know that there are monocytes. Really, mononucleus is the largest cell, and they are only 7% of leukocytes. So there are other kinds of leukocytes too. Macrophage, T lymphocytes, B lymphocytes. So we have to know that there are different types of white blood cells, especially related to the size. Okay, so then the question is, the relevant question, why the capillaries are similar, even smaller than RBCs? Why do we have to have this difficult transit of RBCs through this capillary? And there's an interesting explanation, which I'm not 100% sure it's true, but the explanation here is actually here, saying that, so what's the role of our capillary? That's a business end. Our white blood, RBCs, which contains rich oxygen, has to diffuse their oxygen to the tissue while inside the capillary. So when capillary is small, white RBCs cannot go just too fast. So it slows down so that time is giving enough time to diffuse the oxygen. I think that logically makes sense, but I'm not sure whether this is something easy to prove, okay? It's true that the RBC slows down to increase the time. We call it as a transit time. There's even like a lot of theories on this for gaseous exchange and diffusion of oxygen out of RBCs into surrounding. I think it makes sense, but this is our interpretation, but the proof requires a little more rigorous logic and experimental evidence. So hopefully that makes a sense for Youngmin and others as well. In fact, there's even white blood cells can change its shape. In human blood can also change its shape, because otherwise white blood cells cannot scroll through the capillary as well. And just to make sure RBC size, okay. Okay, so let's move on to a little more of fluid mechanics in our body. So those who have seen this, this is a conventional ways of understanding of the equation. So V equals IR. So this is Q is current, delta P is V, and the other is resistance. And one thing I want to make sure is this is the profile of a fluid flow in a pipe, for example, like our blood vessel. Okay, I think someone asked a question about the sizes of veins and arteries. Unfortunately, that question is a little bit of a issue because this is a cartoon that it never be exactly this way. So this is only like, artery get down to arterial. And in fact, there's a branching out first, second, third order, and then they become a capillary. And that's the business end. The draining of capillary is combined into venar, we call it small venar. So it can also branch in, and then until it becomes a venar and veins and bigger vein and vena cava. So important aspect of here is the pressure aspect. This diagram is wrong, but it tried to convey its idea that arterial blood is pulsatile, but you see your blood pressure is 100 to getting down to almost nothing here, very low in terms of veins. So that's a dramatic down. And let me see. Not. So where comes the majority of pressure drop happens?
I don't know, Professor.
I mean, I'm talking about this graph. So look at that, the pressure, especially artery, there's like 80 to 120. This is we call normal pressure range, okay? And they get down to very, very low to veins. So where occurs 80% of pressure drop in this systemic circuit?
I think it's arterial less.
Okay, great, arterial and
capillary.
Great, right. You can see here it comes down small and large, small and arterials. Okay, arterials it drops and capillary it drops further. So yeah, this is something very much important. Why the pressure drops? Because the diameter drops dramatically, and then it becomes much higher, much more difficult to fluid to flow. So another point out I want to make sure is the other pulmonary artery. This is a little bit more lower pressure. So that's something you have to notice. And let's move on to laminar and turbulent flow. I'm sure you have learned this one from your basic physics course or some fluid mechanics. I already discussed about the definition of Reynolds number, which is a very important parameter. You should know about this. And in fluid, there's some distinction, like 2100. If it's higher, the flow become like this. If it's lower, the flow become like this. So there's a name for that. Thorin. With the low flow rate, the fluid pattern is well big, like this. This is an animal, you inject the dye. Look at this beautiful layered structure. How does it call? Thorin? Thorin.
Yeah, it's laminar flow.
Exactly, yes. Laminar means it's a layer by layer, so it really describes it. What about when the flow becomes faster and faster? Now it becomes like weird shape. Dorian again?
Turbulent.
Exactly. So Dorian, in terms of temporal point of view, what's the difference between turbulent and laminar flow?
Can you ask again, please?
In terms of temporal point of view, over time, what are the differences between laminar and turbulent flow, like this and this?
Laminar flow is really smooth and everything goes in the same direction, whereas in the turbulent one, everything is confused and chaotic, and all the water particles are going in every directions.
Okay, that's good point. In fact, what I want to tell is actually laminar flow can be just, as you say, smooth, so it doesn't look change over time. Turbulent, it keeps changing. So it's an unsteady phenomenon. So in fact, turbulence is good in terms of some areas. For example, internal combustion engine, your car, your gasoline engine actually forced to make turbulent flow to make your fuel to mix well with the oxygen. So that's sometimes we want it, but in our body, usually our body inside is usually laminar. Okay, so that's a point I want to make. Let's keep moving on. So Reynolds number, as I said, 2100, lower laminar, higher turbulent. And I gave you a separate lecture of how to derive this Reynolds number with the solution of Navier-Stokes. This is very, very important. So make sure you understand. And I even provide you the meaning of Reynolds number is the top is more like inertial force versus lower is a viscous force. So low Reynolds number means the flow is more like viscous, like a honey flow. So that's why it becomes more laminar. But if it's more inertial forces are there, like velocity, density, then it's gonna be much more turbulent when I have normal sometimes. So we sometimes use the doctors in the clinic. Okay, let me see, actually, the vessel. So in turbulence is occurring in a vessel or obstruction is happening. So that is really kind of interesting phenomena. We can actually utilize turbulence. So you remember our pressure measurement is based on the turbulence we made when you increase the pressure and then release. And that narrowing down makes the flow faster. So this V is faster and Reynolds number higher, turbulence occurs. We can listen to that sound too. Okay, so that's especially when it can happen, bifurcation. The flow goes and bifurcate, the radius becomes a half, flow becomes higher, and that's where many times this turbulence happens. And another thing is indicating atherosclerosis. So you guys remember there's a disease, atherosclerosis in carotid artery, which can be devastating because it can cause a stroke at any time. And when there are buildup of this atherosclerosis in your carotid artery, the doctor puts stethoscope in your neck, and then they hear, what they hear is like, shh, 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 shh. And that sound is an indication that you have something going on in your arteries. So that sound is called, Doreen again? Doreen, can you guess? This is a little difficult. So if you can't say, I'll give you the answer. It's called bruit, okay? So that's what doctors actually utilize this. Okay, let's moving on. And related to arteries and veins, that pressure difference. I already told you a little bit of this because the vascular and interstitial pressure, the difference is important. And that we call trans-M pressure difference. And let's see. From?
Yes, Professor.
How will you name this pressure difference between this wall of blood vessel?
Transmural.
Exactly, very good answer. You guys probably know this is mural art. Like in Mexico, Latin America, you can find this artwork on your wall, that's called mural. So that's why this is called mural, transmural pressure. And this is important to keep the vessel inflated, okay? And also it determines the rate of water movement out of vessels. So in terms of your vessel, especially capillary, it's very special. It's not actually that easy, just simple pipe, because the role is it actually takes oxygen and nutrients molecules outside of this, from the inside to outside the interstitium where the cells are residing. So the rate of water coming out, okay, and we call this a permeable or measure of permeability out of vessel into the adjacent tissue. So why is this important? Even in tumor tissue, the permeability is different, and usually it's higher, so it's more permeable tissue, and that causes a solid tumor physiology as well. And we have to know about for proper drug delivery. All right, so cyclic variation in pressure is leading to Janice, form again?
Versatile flows.
Exactly, for the arteries, smooth muscle plays because they can contract and dilate in response to physiological needs. So the difference of, big difference of venous flow and arterial flow is versatile flow. And distribution of pressure within vascular system is like the pressure buildup is related to this fluid height. So a good test, I'm not sure whether you have done this before, but this is something, your hand, your thumb part, put the other finger, put it there to feel the pulse. So do it right now, and then not, then I'll see that you guys can feel this. So can you feel this? Yeah. Okay. So what I wanted to try is you can feel this pulse, and then you can lower down, then it's a very strong, and then slowly coming up and up to here on top of your head. Then you will feel the pressure is actually lower. And the reason why, your arm ups and downs is about, let's say, 1 meter, okay? So in the radial artery, this is by itself is 100 millimeter Hg, or water, in terms of water column, it's 140 centimeters. Let's say your arm lower to higher is almost 1 meter, that is comparable. So you will have the differences in pressure buildup. So that's something you need to know. Our body actually responds to this, our heart and all physiological system. All right, so this is probably last part, axial pressure drop in our circulatory system. And we know our heart is like kind of a pumping and releasing, and that pressures are different. Let's say, Jung-Yoon. So when the pressure is high, how do we call that status?
Systole.
Exactly.
Systole.
Systole. And when it's lower?
Diastole.
Yes, diastole. So actually the pronunciation systole and diastole. Okay, so that's something you have to know. So before finishing, I think there's a last question by Janice. Let's see, pressure driven. So is there any chance that pressure in interstitial fluid will be equal to pressure in vessel? If both vessels are values are equal, what will happen? Very, very good question and important question. I think we discussed a little bit of this. In general, intravascular has to be higher to make the blood vessel inflated. But in some cases, this may not be true. Especially, I guess it's more like pathological condition, such as in tumor tissue or when we have an edema. So that Yes, I think I also look for a little bit. So let's go through a little bit. We call it a hydrostatic pressure. And this is a kind of physiologically important. What happens if interstitial pressure is increasing? That will lead, because the pressure difference is low, then we have decreases in microvascular filtration and increase in lymphatic flow and transserosal flow. So in fact, the maintenance of this interstitial pressure is very intricate and very complicated homeostatic process, but it's so important, right? And it's affected by microvascular pressure and lymphatic contractility, serosal permeability. Okay, what happens if interstitial fluid are building up? So it's like edema condition, excessive accumulation of fluid. So our vessel loses a lot more water to outside tissue. What happens? It's detrimental. Why? Edema is forming, and that edema makes the distance bigger. So diffusion distance for oxygen, other nutrients are compromised, which will compromise our cellular metabolism in the swollen tissue. So I guess we can see why swelling could be bad in terms of this aspect. And, and in fact, you know, even in tumors, some tumors are usually higher interstitial pressure, but there are some type of tumor which is more floppy, lower interstitial pressure. From what I know is a melanoma tumor, like black hook sectum, that kills people, and that tumor is a weirdly different interstitial pressure. So using even measuring the interstitial pressure, we actually can consider a biomarker of our treatment is doing well or not, because this is such a complex, but you can ask what determines, what makes this interstitial fluid pressure is like influx to the tissue that is coming from blood capillary filtration, and outflux, that is lymphatic drainage. Okay, and of course the tissue's ability to expand, that we call tissue compliance. And you kind of know the general values. So biologists should know the number. Always consider those numbers. And these numbers are very, very important. But interestingly, some part of hydrostatic pressure is actually normally negative.